to the computer. We're going to share screen. Got this fancy dancy. You had to get a change in the PowerPoint because it didn't allow me to do my old way of doing it. So we have a new format of just changing the background, and it seemed to work okay. All right, test. You got a midterm Monday. I'll post an announcement on what you need to do for that, what you need to study, what the material will be over. I'll also post for the micro on how that's going to be. If you don't remember what I said yesterday, micro is going to be processing a sample from beginning to end, and then you'll get credit for the things you get right, and you'll miss things that you get wrong, and we'll see how that goes through Tuesday and Thursday. Immunology will be on Monday. It will be a handout test for you to do. So depending on how long that takes, but it'll probably take long because there will be dilution problems on there, probably a good bit of dilution problems on there, another problem set probably, so that it'll look like that. So make sure you're good on your dilutions. Also, we had antibody titer, know how you read those and how you set those up. And then we did ELISA, detecting the antigen antibody. You have handouts, 2 sets of handouts on that, and you should be good to review all those. You have questions in there to help you kind of get an idea of where we might be going with questions. So it should be a good midterm. You'll be sitting at your, at the table. You won't be up looking at stuff. I'm not going to do a practical. And then micro, hopefully everything except your strip will be in front of you. But we'll get that together for Tuesday. To start on Tuesday, your midterm grades are due Wednesday. So probably by noon, if I had to guess. So I will put those in. You'll find that grade in Blackboard. What I do is I'll put we put a check by banner midterm, so that grade will be there. I think it's visible if it's not, and you'll know what grade I've transferred. But your course grade will be your grade. So whatever your course grade is, based on that midterm, and based on your assignment so far for the labs. Then the not so good news is that at midterm, if you're not passing at midterm, then you get to come see me. So we start there, then you can go to your advisor after that. But we have to. I like to talk to everybody at midterm if you're not doing so well. There's hope, but there's also being realistic sometimes too. So, but just keep that in mind that you will have to come by to check. If you get a midterm grade that's not a C or better, then you have to come see me. Yes.
I know it's a ways off. Will you have dilutions on your final too?
Nope. No, everything that we do at midterm cuts off for the finals. The finals for lab are not comprehensive. The lecture final is comprehensive. So that'll be from day 1 to last day. So, but lab, we cut it. Midterm is by itself, and then the second midterm is called the final, but it's not a comprehensive. No. But you still need to know how to do dilutions. Don't try to get out of those. Know what you're trying to do. Like I said, those come back. They come back to bite you, and you're like, oh, I forgot how to do that.
I know how to do them in the lab.
Well, okay, we'll see.
Your paragraphs.
That's what you got to prove to me on Monday, that you know how to do them in the lab. All right, chapter overview today. We're moving to 14. This is a pretty intense chapter, so I don't see us getting through this today, especially with a 10-minute late start. So we'll get probably into 1 and 2 of the hypersensitivity reactions, and then we'll save 3 and 4 for Monday, right? But we're about to start cranking up labs that relate to hypersensitivity and then autoimmune, and we'll be measuring certain things with the second half of the labs. So you should feel a little better. The first thing we'll do is we'll do the electrophoresis, start it off second half with that, and that entails a day of work, and then come back and check results. So it should be great. I think it'll be good. I think Usually we try to get, well, I think last year we just got to do, it's like doing logistics or something, but we got to figure out how to get this done with this setup, because last semester we had everybody come in the lab at one time because it was a one procedure method, which means there was prep, you got it ready, and then you did some pouring, and then you did some cutting, and then you did some inoculating, and then you turned it on and you were done. Okay. So last time we had everybody come in, so I'll look and see what logistically how we can do this with our different sections of lab. There might be steps to what you get to do, because that's what it was. We split people up. Like we had the agarose gel people over there with the hot plate, heating it up and putting it in a water bath at 60 degrees, keeping it there. We had our others over here creating slide cutters and well cutters. Then we had our others making up the dilutions that are rehydrolyzing our antigen and antibodies. And then we had pourers and we had inoculators. And not everybody got to handle every one of those things. Okay, so there was some frustration last year. There was at least one student that just turned around and sat down and said, nobody wants to work with me, nobody wants to let me do anything, so I'm going to go over here and pout. Okay, and that's what happened. So I hope that doesn't happen this time, but we will try to assign lab 1 with some activity and lab 2 with some activity and lab 3 with some activity, to that we can do this step by step, and then we'll all come back the next lab and look at the results, and then we'll move on to something else. So I think that's the way I'll kind of seeing the logistics working, instead of putting all 17 of you in the lab together and go, y'all figure it out. That's what I did last year. It was that was bad. Didn't work out too well. It worked out great, except one person got upset and walked, kind of sat down. Everybody else worked together, and and I tried to explain.
Then it probably wasn't you, or the way
I tried to explain that's lab work. You have to have different tasks, and you have to share the task, and and work with others, for cooperation and all that. So that's kind of how that lab's going to go. And then we have plenty more to look at as far as labs go. So we got a full slate of labs coming up, but that'll be after Monday. All right. So hypersensitivity reactions. Maybe you've probably heard of these. Maybe you have some of these. Some of you may suffer from some of these. So we're going to cover them in a way that kind of gets us on track of knowing what's going on. So this very first page, 213, is very important. So of course, hypersensitivity just means we've exaggerated our immune response. So your book says, okay, everything we've done up to here is the body responds to an antigen to eliminate the antigen from the body, and everything should go good. And if it goes good, then the antigen is eliminated and we're all happy. Well, hypersensitivity reactions is we have an exaggerated immune response. It doesn't go as planned. It goes overboard, okay, and it causes harm to the body. So we result in tissue injury and disease. So, a lot of our autoimmunes are because our immune system is hypersensitive and it's overdoing what it needs to do, okay? So we're going to look at the 4 categories. There's type 1, 2, 3, 4. Yeah, those are neat, right? I think we remember that number. We just have 4. And so we're going to define all 4, and then we'll take each one apart and talk about them. Okay, so some of these are immediate. That means they develop minutes to hours after you're exposed. So those that have allergies know that you can't eat certain foods. If you eat a certain food, if you have a peanut in the room, then you're all of a sudden starting to feel all weird, and you start to get short of breath, and you're like starting to panic, and something's going on. You start reaching for your EpiPen, all that good stuff, right? So I think we know how these are going to present themselves, but we're going to take them apart and learn them from the immunology side. We have delayed reactions, and this is, I heard y'all talking about some of the blood bank stuff, and we got some of that to go over too. I was like, oh good, she's already hitting on some of that. I don't have to spend much time on it. But they develop a day or 2 days after. So those are the ones that get us. Those are the ones the patient kind of goes away and everything's going fine. They get home and boom, they have a reaction at home. And we weren't expecting that. And that was a delayed type transfusion reaction. We weren't expecting it, and we have trouble and have to come back. Okay, so some are immediate, and those are good because we know right away that the patient's suffering. But the delayed reactions those are the scary ones because they happen maybe sometimes after the patient's gone home. So we see that as type 1, type 2, type 3, type 4. So in your text, I think we have this. So we have type 1. So I want to go back because I want to just talk about the keys. Okay, so we don't have really the keys up here on the slide, but you have like the introduction to each one is perfect right here on the first page in 213. Okay, so let's take those apart. So type 1, hypersensitivity reaction. Okay, we're going to spend a lot of time in here, but this is what's known, and this is what I want you to group together as a name. So under type 1, we need to know that as also the anaphylactic hypersensitivity. So when you hear anaphylactic, that means type 1. Okay, you just need to put those two words as synonyms and make sure that you get them associated that way. It is, in these reactions, there's an exposure to the antigen. That makes sense. We're immunologists, we know that, right? There's an antigen exposure. And the production of the specific immunoglobulin, IgE. Okay, we hit on IgE and we talked about it, what it looks like, and kind of its population is kind of low. But when we talk about anaphylactic type 1 hypersensitivity, IgE is the player, okay? That IgE antibody binds to receptors on the mast cell and the basophils. So the IgE captures the antigen, okay? The antibody captures the antigen, and then it has that FC region down there that attaches to these mast cells and basophils. Once that is bound, there is a degranulation of those cells, and chemical mediators are produced, and there's allergy symptoms. So we call this a type, oh, sorry, that's it, right? No, cell bound regulation. Okay, that's it. Sorry, I was heading to number 2 without period. So period right there, okay? So IgE attaches to the antigen, FC region attaches to the mast cell basophils, degranulation occurs, and we'll have pictures, okay, coming up. Now to 2, type 2. Type 2, also known as antibody-mediated cytotoxic hypersensitivity. So type 1 is anaphylactic, type 2 is antibody-mediated cytotoxic hypersensitivity. And you're going, well, isn't IgE an antibody? Well, it is, but it's not part of type 2. Type 2 is IgG and IgM. You know those. They're the major players, especially IgG. They react with the antigen on the surface of the host cell. So the antigen is not just freely out there and captured. That antigen has made its way to a host cell, and these antibodies then find it and attach, okay? And this can lead to cell damage because we know complement. You've already had complement. The reason we spent so much time in complement, it was because you now know complement mediated can lyse that cell that the antigen was attached to, which the IgG and the IgM came up and found. Y'all were talking earlier about some of this in the last class. So once that happens, there can be dysfunction of the cell by blocking the binding ligand of the surface receptor, and then there's overstimulation of the cell's function can happen too. We're going to take that apart with type 2. But that is type 2, antibody-mediated cytotoxic hypersensitivity, not IgE, IgG and IgM. Okay. Type 3. Type 3 is also referred to as complex-mediated hypersensitivity. So we got anaphylactic, we got antibody-mediated cytotoxic, and now we got complex-mediated for type 3. This process involves IgG and IgM too, just like 2 does. They react with soluble antigens, not antigens on the cell, but soluble antigens to form small complexes that precipitate into the tissue and activate complement. Okay, so these are the things when we think about autoimmunity. We think about those complexes, and when we say, hey, your kidneys aren't happy. Your kidneys are filtering that blood. Your kidneys now have these big complexes due to this reaction of the IgG and IgM on the antigen. Have taken it into the kidney, and the kidney now has these deposited in it. Okay, so these complexes end up in an organ system and stay there. So the kidneys have to try to clear that, and they get bombarded and they get damaged, okay, by these complexes. So recruitment of the neutrophil to this site causes the inflammatory response and causes injuries to that tissue. So that complex of IgG onto the antigen floating around, bound up, went into the system, went into an organ system, and all of a sudden neutrophils find it, show up, and start wrecking havoc into that tissue, causing damage. Type 4, hypersensitivity, differs from all the other 3 because it is sensitizing the T cell rather than the antibody. So if you hear which one of the hypersensitivities involves T cells, specifically, it's 4. They all can have T cell activity, but we're focused on T cell, which is cell-mediated hypersensitivity, is the other name for it. Okay? It involves the release of cytokines that induce inflammation and tissue damage. So types 1, 2, 3 from this slide, right, are immediate, and type 4 is delayed with that T cell. Okay? All right. So hopefully that's got us on the same page as we move forward into each one of these. And we'll see. I mean, your book, all the chapters in this text have been pretty easy to, it's not hard, but this one seemed like, oh my gosh, we know a lot about type 1. This author spends quite a bit of time on type 1 hypersensitivity. So if you've read ahead, you're like, oh my gosh, yes. And got some really good pictures. And they're not due to graphic material. There's going to be red skin, so you're okay, unlike micro. So type 1 hypersensitivity, anaphylactic hypersensitivity, good. I've got 2 or 3 test questions already taken out right there. You can link those 2 words together. Typically thought as allergies, commonly occur within minutes of exposure to the allergen. And I apologize, I tried to, with this new format, I came up with these PowerPoints. I couldn't change the G back to small. It wouldn't let me put small letters. So I got big immunoglobulins, okay? I got big IGs. So sorry about that, but if you're like, that looks weird, I've never seen it with a big G in it, but yes, I couldn't switch it for some reason. And it was to the point, I was very, very upset last night because I went in to do the PowerPoint. I usually do it the night before, and I was like, oh, I'll just go in and edit my 30-something slides and get it ready and put it on everything, get it ready to go. And I went to change them, and it's like, it's not there anymore. Like all of a sudden my PowerPoint program decides to update or something and take away my customizing, with the blue and the orange color back, the banners and stuff. I can't find that now. So we might just have to go to water droplets or something this one for a while. Anyway, the key components, IgE, okay? Don't forget that. This one is, you didn't hear about IgE anywhere else except for this one, type 1. Mast cells and basophils and eosinophils. Okay, so you're in that right now, right? You're in hematology. And all of a sudden you get a slide and it's all these eosinophils. And you're like, well, I thought eosinophils for like big parasitic worms and elements and hypersensitivity reactions, right? And we also know basophils play a role. So one thing I like to tell the story was one of the coolest, cases I had early on in my career. We did an ER patient, a little kid, infant, basically, and I drew the blood and I took it and ran it, and all of a sudden I got these eosinophil count just went up to about 30%. Like that's never supposed to happen. Now you're supposed to be over like 5 or 6. All of a sudden it's like 30% of my white cell count. So I go back to the pediatrician and I go, got something for you. And he looked at it and says, wait here. So it's kind of one of those, like, wait here. So we had curtains in our ER, separating the beds. So I curtain pulls and pulls back. And so I hear him go, and you cannot smoke in front of this baby anymore. So it was the cigarette smoke. These parents were smoking with the baby in the house or in the car, but he was giving them the old lecture of, this baby's getting exposed to allergens. This baby's getting exposed to cigarette smoke. This baby is not supposed to be running up eosinophil counts on their CBC. So
Is that always the case?
when you see it jump like that, it could be allergies, yes. They increase. So some kind of like hay fever season, that kind of thing, you can see your eosinophils kind of start to move up. It's not always helminth parasitic infections. We don't get very many of those. But we're going to talk about where this happens in this whole chapter here. So here we are. Here's our, we have stages. Okay, so on 214 you got 4 pictures. You got type 1, type 2, type 3, type 4. So that's kind of a summary of what's going on with pictures. So I like that. But we have steps to this type 1. Okay, so we have a sensitizing or sensitization phase for type 1, where you're going to see that we have to sensitize. And so how does that do? We have antigen presenting cell processes the allergen. Okay, so here is the allergen, little green right here. Okay, that's a piece of peanut. That's a piece of dust. That's who has allergies. What else are you allergic to? Cinnamon. That's a little piece of cinnamon.
Cats.
Cats. That's dandruff from an animal. Dandruff. Anybody else?
Mold.
Mold.
All of the above.
All the above.
So my brother, really random, has a major allergy to propylene glycol. How would you get exposed? It's a preservative.
Okay.
Like
No, what we're saying is this is not, don't take these as you got to get exposed to these somehow, right? This is just these antigens come in, and whatever your predisposition to allergies is, we're pretty sure it's genetic. Okay, we're pretty sure that you have a predisposition to develop allergy, especially peanut allergy. Okay, it's just isn't a thing that everybody's allergic to everything, and somehow people work through it. But we do have predispositions. So the antigen presenting cell is going to process the allergen, and then it's going to present it to the T helper cell. Now the difference here than the normal immune response to that is that T helper 2 is now the major player. Okay, and that's the problem with type 1 hypersensitivity, is that we usually have equal responses and regulations and help from helper 1 and helper 2, but with allergies, helper 2 takes over. Okay, so the balance of the immune response is weighing over to helper 2 cells, and helper 1 is out of the picture. Okay, so that's the problem. So when you say what is the main concern, is that helper 2 cell is playing the major role. So the helper 2 cell induces the production of allergen specific IgE antibody. How does it do that? The helper 2 produces 2 interleukins, 4 and 13. That's the signals to the B cell to what? Go through its maturation process. Now it's sensitized the B cell. The B cell turns into a plasma cell and starts spitting out IgE antibody. Right? Everybody good with that? Y'all should be, because that's where we spent most of that first half of this semester getting ready for this lecture. So this wouldn't be foreign language to you, that you would understand, if not all of this, at least enough that you're not like, I have no idea what he's talking about today. I don't expect to see that in your eyes or your faces or your voices today. Because we made a point. This is the whole point of last year versus this year was I wanted to spend more time getting ready for this. Okay. I didn't want this to happen, this lecture to come in and y'all go, I don't have any idea about interleukin 4s and 13s. I don't have any idea about how the B cell is stimulated. And I definitely don't know how the plasma cell starts to spit out antibody. Don't know how that happens. Okay. But what you see now is that IgE is sensitized. The IgG is there. Okay. And then who's this over here? Who's this guy? Who's this over here? We got an FC, and that's an epsilon R1. Who's this over here? That's the basophil. It's a basophil or it's a mast cell, one of the two. Okay, now it has these receptors for that FC portion of the IgE. So this increased sensitizing of the difference, where we got this, all this IgE starting to be produced. Now these mast cells start to link up, and when they link up, and they see that allergen again, then that's when we have the response of they degranulize and they do their thing. Once they release, basophils are a lot better at releasing that stuff than the mast cell. They got more pop. Okay, so they're going to release it, and that's what we're going to look at here. We're going to see these products, okay. The IgE binds to the FC epsilon region R1 receptors on the mast cell or the basophils. Then we have to go to activation. So I think the next step, we're going to talk about the activation step. Okay, so we're just pick it up, same picture, nothing's changed. Same picture. The allergen cross-links adjacent cell-bound IgEs. You see that cross-linking? Just basically here, the two arms of different IgEs. Now we got a cross link here with the activation, with picking up that allergen. The mast cell and the basophil degranulate, meaning they let out what they're holding. Those are chemical mediators. They're released. They bind to target organs, and the allergy symptoms arise. Okay, runny nose, cough, watery eyes. You know the drill. You've had allergy. You know what's happening here. Okay. So that's the activation into the symptoms, and then we have this list. So these are all of our mediators of type 1 hypersensitivity. No, these are the antigens. Sorry, these are not the mediators. Sorry, got ahead of myself. These are the allergens. Pollen, mold. I think we mentioned all of these, right? Animal dander, dust mites. We didn't think of that one. We didn't talk about insect venom. Nobody was allergic to bee stings, wasp stings. Nobody?
Mosquitoes.
Don't know yet? Mosquitoes.
Yeah, you
Mosquitoes don't have, I mean,
Is
don't
that
have
like,
venom.
is that like an allergy
But not
reaction, like
constructive.
mosquito bites?
Yes. I think so. I mean, but is the mosquito, what is the mosquito spitting back in your bite? It's drinking your blood, but what is it leaving behind? You don't know. So you might be allergic to that. Some people are really allergic to mosquito bites. Not that they have anything other than a welt come up, but that's on a reaction. That's what we're looking at. So yeah. You don't know if you're allergic to bee stings or wasp stings, never been stung. Well, let's hope you're not.
I know.
Right? That'd be a bad time to
Because I'm
find out when
allergic
you do
to
get
everything
stung.
else. That's what I'm scared of.
You never like walk through like grass with no shoes on and pick up a bee that just decides to latch on, and you never had a wasp come at you and pop you like pop, and make it burn like crap over with a red wasp. Don't ever, you know.
I grabbed out of a rail once and like grabbed a hold of it.
Yeah, I've done that too. Like reaching up somewhere and all of a sudden you think you grabbed a hot wire or something's burning and it's a wasp on your hand.
Okay.
Okay.
So like the whole black widow thing, is that what kills people if they're allergic to them?
That's yeah, it's yeah.
Or is that
When
not
we're
really?
thinking bee stings and wasp stings, black widows are different, kind of a little different, but I will say I think
Because like people always
Not
ask
up on
me
my black
allergic
widow
to them,
venom.
and I'm like they're poisonous, so I would think they hurt.
but I am up on my brown recluse venom, and that is a tissue. That is one of those where the tissues start to be attacked.
So
Brown
it would
bite.
be an allergy.
So it kind of melts away. Peanuts. Anybody allergic to peanuts? Zoom, y'all can chime in. I'm not trying to leave y'all out. Shellfish, milk, dairy products, eggs, right? Reason we got to worry about the flu vaccine, right? Because it might contain egg products, and some people might be allergic to eggs. So they can have an allergic reaction to the flu vaccine. So if you're allergic to eggs, they don't want you to do the flu vaccine. Certain drugs, penicillin. We have patients that come in all the time allergic to penicillin, which is a mold, right? So, and latex. Yay, I can tell a latex story. Yeah. Latex is our, you guys, you know, you have nitrile gloves in the lab for a reason. And that reason is because we have latex allergies in healthcare workers up to 30% sometimes. Okay, sensitizing, you wear the latex glove over and over like I did, and all of a sudden you have reactions. So I have two examples of latex. We always do what? When you go in as a phlebotomist, we say, are you allergic to latex? Well, let's take the phlebotomy story first. So we usually think of latex as I'm wearing gloves. I have nitrile gloves. I'm not worried about you and latex allergies, right? And we got to make sure our tourniquets aren't latex, right? But you know what else you got to worry about with latex? And I'm going to surprise everybody. Hmm? Patient. No, something we use. Y'all might not be using it downstairs yet, but you've heard of Coban? That wrap? They wrap your arm with it after you donate blood or draw blood.
Oh, yeah.
Coban, that wrap has latex in it if you don't get the non-latex version of it. So I was like, oh, really? I've been putting that on patients and never thinking about that having latex in it. Yes.
The Red Cross thing that they use?
Yes.
It has it?
Yes. If they don't use none, they make it without latex, just like gloves. But if you're not careful, nobody's pointing that out. They'll send you a whole box. And then you read fine print, and it says this product contains latex. And you're like, really?
Is it common to be allergic to vinyl gloves?
I haven't heard of that. Like nitrile, no. I haven't heard of anybody being allergic to synthetic gloves, no. Not yet, but we don't think so because latex is it's a natural rubber that definitely could be the engine part. So I have another story with latex, two more stories. So the next one is I have a nurse comes down to the lab and goes, what is wrong with my hands, right? Oh my gosh. So the doctor looks at her hands and goes, well, I think you've gotten into something like a fungal infection. So we need to culture your hands. We want you, how are you cleaning your hands? And she's like, I'm using betadine. Like I'm betadining my hands, trying to get off whatever's on my hands causing them to be red. I'm trying to get that off. So the doctor actually she goes to one doctor and she comes back and tells us that this doctor wants to take a biopsy of her skin out of her hand and have it looked at. Like she's going to take a plug out. I said, would you go to my dermatologist, please? Because he's old school and I don't think he'd be taking plugs out of your hand trying to figure out why your hands are red. She goes to the doctor, right? My dermatologist walks in and goes, what's your profession? She said, I'm a nurse. He said, you got latex allergies. That's all. I don't need to do any more for you. Just quit wearing latex gloves. She did. Of course, her hands got better. They quit being red and raw and quit putting betadine on them and alcohol and all this stuff, just making it worse. So that was one, but that made sense because it was contact here on the hand, wearing gloves every day. And in lab, we pretty much put them on, constantly. You are in lab with me an hour and a half. So I have a personal case. So I'm going to Camp Bear Creek. Does anybody know where Camp Bear Creek is? Down by Mariana. So I got a whole van full of kids I'm taking to Camp Bear Creek for the weekend. And my eyes are swollen. It's like 1st of May. My eyes are swollen up like I look like I've, gotten into something. I'm wearing my glasses and I don't know what it is. And I'm like, I just know I can't wear my contacts. I'm wearing glasses. So I go back to the lab and I go, those girls at the microscope, they're leaving makeup underneath the eyepiece, and I'm probably putting that up against my eye, and I'm probably allergic to makeup. So I'm gonna clean those and make sure it's clean, right? Not getting any better. Then my skin starts to blister there, and it actually peels underneath my eyes. And I'm like, oh my God, I'm free. I'm allergic to latex. So it wasn't that I was wearing the glove on my hand. My hands never did anything. But I wipe my eyes quite a bunch. So I'm taking whatever I had on my hands, putting it up on my eyes. Wasn't a microscope. I changed over to nitrile gloves. Story. No more swollen eyes for me. So as a healthcare worker, a lot of us over the years have become sensitized to latex. Now, nothing respiratory, nothing where if you brought it into the room with me, I would have a reaction. But it is something that we link to long time exposure to the allergen. So I got all those stories out of the way, and we still have like 10 minutes. Okay. So this is how the clinical manifestation of type 1 looks like. We have rhinitis from hay fever. We have asthma. And asthma, from your, if you haven't read your text, you're going to find out, love these little things, that this is Greek for panting. So asthma is a Greek word meaning pant. So we get a lot of our medical terms from Greek. Food allergies, urticaria hives, the wheel and flare. So we think about what? We think about our TB skin test that we do where they go sub-Q with the allergen and say, well, did it wheel and flare? Did it raise and red, right? So it looks like you got this kind of wheel and flare, which is a raised off the skin and starts to flare out. Eczema, if you watch TV, everybody suffers from eczema evidently because there's a drug commercial for eczema, every other one. I don't know why that is. I don't know very many. I guess it's a problem I don't know about. I don't know. But it seems like a lot of people suffer from eczema. Systemic anaphylaxis, this is where we worry about it getting introduced into the body, not just on contact out on our bodies, but we actually get it systematic, and that causes big bad problems and can be a fatal reaction. So we think of anaphylactic shock, okay, from the bee sting because we didn't have our EpiPen with us. The allergy, the peanut allergy. My kid could have died today at elementary school because your kid brought peanuts with him, or your whatever they brought has peanut in it and you didn't know. And you brought a peanut butter and jelly sandwich to school. All that stuff, right? This is why we have, those rules is because we have those sensitizing allergies. All right. So I think we kind of missed this part. I was kind of disappointed. I guess I'll come back here because I missed table 14.1. I think this is important. I think we're smart enough in immunology that we need to know 14.1, which is the primary preformed mediators. Okay, this is what's inside the basophil. It's what's inside the mast cell. And we've talked about them just kind of in passing. Yeah, they got granules, and y'all are in histo. Y'all know what the granules are. Y'all going over granules? What they are inside the basophil, and maybe. No?
We talked about how they would look.
How they look? Well, those mediators, granules inside those cells are histamine. Yay, right? So we now know why we do antihistamine when we have allergies, right? Take drug, take Benadryl, right? That's an antihistamine, which is supposed to what? Neutralize the histamine granules that the basophil is laying out because we have seasonal allergies. Heparin, y'all are all familiar with heparin, I hope, right? Heparin does what? Heparin keeps our blood from clotting, right? It's part of the coag cascade that you're going to learn sometime, if you hadn't already. And we know that we have heparin in some of our tubes. If you've done phlebotomy, remember we have lithium heparin in the green top. Does that sound familiar? And so it's an anticoagulant. We have eosinophil chemotactic factor of anaphylaxis. That's our ECF. We have neutrophil chemotactic factor. Okay, so what is that? That's bringing those cells in. That's bringing those to the areas. We have proteases, break down proteins. We have tryptase and chymase. And those are kind of the mild mediators. I know they call them primary because they're preformed, but the secondary ones are more potent. And these are the ones that are newly sensitized when you have a reaction. Those are prostaglandin. Those are leukotrienes. We have many of those. We have B, C, D, and E. We have platelet activating factor. Yay, y'all know that, hopefully. And cytokines, interleukin 1, 3, 4, 5, 6, 9, 13, 14. We see interleukin 6. We always like to see interleukin 6, right? That's our COVID-linked interleukin. 16, tumor necrosis factor alpha, and colony stimulating factor. Okay, so we missed those, but we didn't want to bring those in because those are what really makes this happen. All right, so we're going to get to, I think we're going to do the treatment, and then we will end because it's I got 9:48. What do y'all have? We got 9:49 now.
Yeah.
So we'll do the treatments and we'll pick up with the testing because I don't want to short the testing because I don't know if you've ever done that. I haven't, but anybody been through this kind of testing?
I did it in August.
Yay, all right. So
It was not fun.
did y'all take pictures? Did y'all have pictures of your back? Or, no?
My mom does.
Your mom has pictures? All right. But treatment for type 1 hypersensitivities, avoid the allergen. Avoid cats. Avoid peanuts. Avoid bee stings. Avoid shellfish, right? We can do that. And we know sometimes we can't. We can take drugs. We take antihistamines. We can do bronchodilators in case all this stuff is starting to, make us get a little short of breath, kind of getting hard to breathe. We feel like right? Somebody eats peanut butter and they're like, I just get a little tight up here. Everything seems to be, I don't know if I can get any air through that. It's kind of the feeling that people describe. I'm not allergic to peanut butter, but mast cell stabilizers, steroids, and then our epinephrine. Okay, that's our emergency EpiPen that will allow this to reverse, to allow us to open those back up and give us a little pop to get us through this reaction. We can do monoclonal anti-IgG antibody. We can go after that IgE. Okay, that IgG has been sensitized by peanut allergen, and then it's floating around. Then we can treat the patient with the anti. We can use allergy immunotherapy. Administer a gradual increasing dose of the allergen even. And what that is, here is an immunotherapy. We have the name. I'll pull the name before we leave out of your chapter. But this is new. I was in part of where I worked at one time, 1996 to 98, I think. Does that sound right? 1996, 98. I was working at the UAMS Nutrition Center. And I was friends with a pediatrician that worked there. I grew up with her, and she had gone straight into research, and her research is being published. You look up peanut research at UAMS, and what they were doing was they were finding the amounts of the allergen that would allow you to become desensitized to the allergen. So what they were doing was giving small doses to kids of peanuts if they were allergic to peanuts to reverse the big boom that the type 1 hypersensitivity reaction would work. So this was new. This was like she was doing this. So they were desensitizing the patient to the allergen by giving them small doses of the actual allergen. So that's what administered gradual increasing doses of the allergen to kind of desensitize that one big womb of, oh my gosh, got an anaphylactic reaction to peanuts. And they work their way out of the allergy. So that's new. But I did want to point out the monoclonal, that antibody that was here given. It is called, anybody find it? Does anybody follow along? Omalizumab. Okay, so the antibody at the end is AB. but it's OMA, OMALIZU mab. Omalizumab is the monoclonal therapy to treat. It's the anti-IgG. So it goes after the IgG.
Wait, where do you see that?
I'm on, sorry, 218, the treatment of type 1 hypersensitivity. All right, that's where we're going to end today. So anybody have questions? Zoom have questions? Want to make sure we have a really good background on these type hypersensitivities. So I don't have a problem doing another day of lecture. And then you're going to get some blood bank in type 2, and then type 4 is pretty basic. So it's not a whole lot. Like, they spend a lot of time on type 1 in this chapter, so that's why we still spend time on it. But once we get through 1 and go into 2, then it gets a little easier to finish up. We'll finish up Monday. All right. Y'all have a good weekend. I'll look for my post about the midterms and help you give you some pointers on what we're going to be covering. And we'll see you Monday for lecture. And we will have lecture. And then we'll have lab midterm in the afternoon. I'm going to stop the share. In the lecture. See you later, Zoom.